All right. Hi, guys. Welcome to AP Biology Cram preview number 2. I am so excited that you guys are all here. Um, it's going to be a fun night. I hope you guys are ready. We have 1 hour to give you lots of tips and tricks and ways to get as high as the score as you can on the AP Bio exam. So welcome. In terms of how to communicate with us, we have the chat feature, so you guys can go ahead and ask us questions in the chat. And I have 3 amazing student hosts with me today that are going to be presenting with me and then also be helping answer your questions in the chat as well. All right, so you are going to want to have maybe a piece of paper with you so you can take down notes, because we're going to give you lots of fun ways of getting like really good ideas for your test. All right, so as always, you're going to want to follow us on our socials, so make sure that you're following us. Fiveable has amazing resources to help you prepare for the AP Bio test coming up next week, which is crazy to think about, but you guys can do this. Here is the cram session that we have starting on Friday. It is 5 days in a row, 7 to 10 p.m. Eastern time. You guys are all on different time zones. And we're going to be going over different parts of the exam. So on Friday, we're going to be doing units 1 to 4. And then you guys can see the rest of the schedules right there. And then we're going to end the night before the big day together with a giant grand finale of everything and anything you want to know to get yourself ready for the AP Bio test. So before we get started, I want to kind of introduce my amazing host. So I am Sabrina Shumate. I am presenting from Virginia Beach, Virginia. I'm an AP Bio teacher. And I'm going to have my student hosts introduce themselves. They're going to share their name, where they're presenting from, and they're also going to share with me their favorite ice cream, because I do love some ice cream.
Great. Well, I will get started. Hi, my name is Madison, and I am a high school sophomore in Illinois. And my favorite ice cream flavor is mint chocolate chip. A lot of people do not like that flavor, but I do.
Hi, I'm Rashi. I am a college freshman and I live in Tampa, Florida. And my favorite ice cream flavor is probably cookies and cream. I know it's kind of basic, but honestly, I feel like it just brings me so much joy. And it's not as chocolatey as chocolate, but it still has more flavor than vanilla. So if you also are a cookies and cream fan, please drop that in the chat.
Hello, I'm Samhita Paula. I'm from Dallas, Texas, and I'm a high school senior. My favorite ice cream flavor would be chocolate.
I love it. If you guys would like to share your favorite ice cream, feel free to put it in the chat as well. I'm a Cold Stone type of a person, so I love all the toppings and everything. And I would say my favorite ice cream is, I actually like coffee ice cream. I know not a lot of people like coffee ice cream, but I do love coffee ice cream. So again, feel free to share with us if you are an ice cream lover. All right, so what you guys can see on the screen now is kind of a detailed preview of all the different topics that we're going to talk about each of the CRAM sessions if you choose to attend them. And here is our agenda for the night. We're essentially going to recap last session if you were unable to attend or view the session. And then we're going to kind of go into science practices and formula sheets and sample problems. And that's going to be kind of the focus of our hour together. All right, I'm going to go ahead and hand it over to Ms. Madison.
Great. So sorry about that. Hi again, Madison here, one of the other student hosts, and I will go over a quick recap of rehearsal 1. Rehearsal 1 was actually with another instructor named Ms. Kari, who you guys will be seeing within the next week. And we also had 2 other student hosts during rehearsal 1. We had myself, Jeffrey, and Joyce. So 2 other student hosts were involved as well. We began rehearsal with some information on the upcoming AP exam, such as the details, the format of the exam, and most importantly, some of the tips and tricks for those exams. And according to our slides as well, Ms. Shumate will also be discussing that as well, so do not fret if you're unsure of the AP format. After some quick review of the exam, we went straight forward to working our skills on multiple choice and free response questions. Surprisingly, we actually used all of our time last week on practicing FRQs, multiple choice, and mostly around the units of self-communication, hereditary, etc. And if you're interested actually in practicing your skills through multiple choice and FRQs, within the meeting, I will be sure to send over the link that actually shows the whole recap of rehearsal 1, the whole video showing the questions, and also as well as the AP format for the exam. And so now let's get started with tonight. Next slide, please. So we would love to see in the chat of where you guys joining from. Let us know. And actually, why I did say I was in Illinois, I'm not sure if our other students said where they were. Did you guys say where you guys are coming from?
Yes, we did.
Oh, perfect. Sorry. Oh my God, someone else is also in Florida. Isn't that cool? Oh my gosh. New York, Georgia.
We got a world. I love that.
That's amazing. All right, please keep going in the chat, but we'll go to the next question really quick before we get started and before I send over Ms. Shumate to discuss the exam information. What is your guys' favorite unit from AP Biology? Personally, I enjoyed actually unit 5 on hereditary. I honestly just loved looking at pedigrees and answering questions regarding our genotypes, phenotypes, our chromosomes, etc. I was just a huge nerd on that. But actually, Samantha and Rashi, what was your guys' favorite unit currently from AP Biology?
Mine has to be genetics. I really loved Punnett squares. I was like, wow, this makes so much sense to me, very straightforward.
Yeah, I also like Punnett squares and pedigrees. They were like little puzzles.
Exactly. For me, like at least for my class, we did like a lot of projects revolving on genetic disorders, and we also had to make like our own pedigrees, which was kind of amazing. But yeah, I definitely gained like a huge interest in genetics as well.
And Natalie, you don't have to know it by heart. Which one was your favorite, dear? No worries that you don't know unit 1 through 4, whatever it is. What was your favorite in general? I would have to say that my favorite is all of them. I love every single one of the units.
I see that we're like very expanded. Like everyone has like it's like with across the board. A lot of people are interested in like mostly all the units. Not everyone, but there's a huge like range. Great. However, I will now send this over to Ms. Shumate to let her discuss the AP exam format, but please keep going in the chat.
All right, thank you so much, Madison. Good to know that we are kind of all over. It's interesting to me to know the different time zones. So it is like 8 o'clock at night where I'm at. So I've had a very long day, and some of you guys are just kind of starting your day. So in terms of the exam format, your test is going to be 2 parts. You're going to have 60 multiple choice questions, and then you're going to have your FRQs. And each part is going to be worth the exact same percentage, 50%. You're going to have 90 minutes to do the first part. You'll take a break, and then you're going to have 90 minutes to do the second part. Okay. And then your score is going to be kind of combined together. And so what we're going to focus on tonight are the science practices that you are going to be tested on. So the AP Bio exam is interesting in the fact that it's not recall. So you're not going to have something that's just going to say in 1942, what happened here, like in your other subjects. It's going to be more of case studies and models and graphs and diagrams. And they're going to ask you questions based off those case studies. You will have probably never heard of any of them, but that's okay. What you're going to do, and my little trick for you, is focus on the model or diagram that they give you, and you'll be able to answer most of the questions. I know that A push recall was traumatic. I'm sorry, but AP Bio is not like that. Thank goodness. So it's going to be the application of the science practices. So you can do this, okay? Even if for whatever reason you were absent a lot for a particular unit, it's okay. You can kind of focus your studies on that and remember that it's going to be the analysis of data tables and graphs and models and case studies, okay? You can do this. You can totally do this. All right, so let's talk about the science practices itself. Okay, so there are 6 science practices, and I am sorry if it's really small on your screen. You guys can look it up. It's in the curriculum guide that we have for AP Bio. But there's 6 things that you guys can do. It's going to be, can you look at a graph and form a conclusion? Can you justify your answer from the graph? Can you try to explain why you think you got this result? So these are all things that you guys did throughout the year already. Can you do some sort of data analysis? Okay, so it's nothing that is going to be your kind of be like recall type of questions, which is kind of nice because that's a lot going on. So you are going to have to do a little bit of math, but you can do it. It's okay. Math and science go together. And luckily for us, you'll be given a formula sheet. Thank goodness. You don't have to memorize a single formula. Woohoo. And so on your formula sheet, what I would do is I would look at the formula sheet that we have on here. And again, this recording will be available for you after we're done. Or if you just Google AP Bio formula sheet, you're going to see all the formulas that you might come across. You might not have all the formulas, but there's going to be all the formulas in one place, which would be nice. So if I were you, I would kind of look at the formula sheet and at least know how to use those equations. For your AP test, you also get to use a calculator. So I would make plans now to figure out what calculator you can bring to the AP Bio test. You will not be given a calculator, and if you don't have one, that's not going to be the best thing ever. So make your plans now to bring your own calculator, figure out from a friend. You know Murphy's Law, if something bad is going to happen, it could happen. I would have extra batteries, and that way my batteries are set because you don't want to have a calculator not be able to work and then they can't replace it. You want to make sure you have 2 pencils because you will use pencils for the multiple choice, and you want to have blue or black pen for your FRQ. You want to make sure that your calculator is going to be approved. And so I have the tiny URL at the top. I just made it tinyurl.com, AP exam calc policy, if you want to look it up. And it has all the lists of the calculators that you can use. So you can use 4 functions, no big deal. I would definitely use a graphing calculator. What they're really wanting to make sure is that your calculator cannot access the internet. That's like the big no-no for what's going to be approved or not. So most of the ones that you guys have are not going to access the internet, so you don't really have to worry about it. But if you just want to make sure that you can use your calculator, I would check that policy, and that way you guys have it. All right, so one thing that I do want to kind of go over tonight with you is most of the graphs that you're going to see are going to have what are called error bars. And so if you look at the graphs themselves, you kind of have one graph over to the left is kind of like sideways looking. You have one over to the right that's kind of normal what you're looking at. But what you'll see at the top is you have these error bars. And so whenever you see these graphs, these are kind of our bar graphs. Where the bar graph kind of goes up represents an average, right? So if in this experiment, I have enzyme activity, I have a control group, and then I have a treated group, my experimental group. And so when I run my experiment, I might have 100 trials. I do not want to plot 100 different data sets. That's like crazy talk, right? So what I'll do is I'm going to go ahead and I'm going to just graph the average, right? Because the idea is for us to figure out a trend. If I have a bar graph with 100 bars, I'm seeing it's too much, right? So I'm going to take an average and say, on average in my control group, I have this much enzyme activity. In my treated group, I have this much enzyme activity. And so these error bars are kind of showing you the spread of the data. So I have a little data above, a little data below on both of these. So whenever you see error bars, and you will see them in the multiple choice, you're also going to see them probably in the FRQ. Most of the time, they're going to ask you to form a conclusion using this information and then justify it. And so the method that I teach my students, and I want to teach you, is called CER, which is claim, evidence, and reasoning. The claim is your conclusion. So anytime they give you a graph and they say to make a claim, they're basically saying make a conclusion. And like for this one on the left, you can make multiple conclusions. there's more than one conclusion. You could talk about just within this first team, or you could talk about this versus this, or overall, okay? It doesn't really matter what the conclusion is as long as you can justify it, and that's the E, that's the evidence. And so what you'll say is according to the graph, the control group has a higher enzymatic activity. So that is a conclusion. I usually tell my students to say, according to the graph. Then you justify it. And this is where you're going to talk about the results being significant or not. So anytime you see error bars, make sure you talk about significance, okay? So if the error bars are not overlapping, that means that there is some significance, okay? If you have some of the error bars overlapping, then we would say there's really no significance, okay? So like, for example, for my control group, let's say that I had, a bunch of numbers that were like, I can kind of look at like 140, 142, 143. And then over here on the treated group, I almost got the same numbers. And we could tell that because the error bars are overlapping. So really, I kind of got the same numbers. So the results are not significant. But if I have totally different data and none of it overlaps at all, then your results are significant, okay? So when dealing with error bars, remember that you always want to talk about significance. And if those error bars are not overlapping, then those results are significant, okay? What you got from one group is different from another. But the more overlap of the data that you have, then that's showing not significance. Most of the FRQs that I've ever seen have been analysis of the error bars. On one of the skills and drills, we are going to do some practice of creating the error bars. Remember that when you do the error bars, you do 2 standards of the mean. And from all the ones that I've seen in the past tests, they usually just give it to you and you just have to plot it. So it might say like the average for the control is 145 with the 2 standard error of the mean being 6. So that just means go up 6 and down 6. That's all that it means. Okay, so just remember when dealing with error bars, see it and know that's talking about significance, and that will get you a lot of the points or whatever the case may be. All right, so now we're going to go ahead and move on to focusing on science practice 6. So at this point, Samantha is going to kind of take over, and she's going to be your expert on teaching you some tips and tricks for science practice 6.
All right, let's go to the next slide. Okay, so this is a problem from the 2021 AP Bio exam. And it basically says that scientists conducted experiments on this krill species to detect the changes on HSP genes when the krill are exposed to temperatures above 4 degrees Celsius. So first they're going to be at 4 degrees Celsius in seawater, then they're going to be moved to 10 degrees Celsius seawater for 3 hours. And then there's a 6-hour recovery period in which they'll be placed back into 4 degrees Celsius. And here's the graph that we use. So as it shows that the key has 3 mRNAs that we'll be basically using to answer the questions, and the y-axis is the average mRNA concentration, while the x-axis is the time. And the questions will be on the next slide. So here are the questions. They're basically straightforward, and we'll be looking at them on the next slide. So we'll be starting off with answering part A and B. Part A is identify the HSP gene mRNA that has the slowest rate of concentration increase in response to heat shock treatment. And by identify, they just mean just answer the question. No need to elaborate. It's usually a 1-sentence answer. And for part B, it's describe the trend in the average concentration of mRNA1 throughout the experiment. So for this, you'll probably need some background knowledge, but you're basically going to be referring to the graph. And I'll be giving you guys 1 minute to answer these 2 questions and begin. Does anyone need more time? Okay, here's what I answered for part A and B. For part A, I said the HSP mRNA-3 has the slowest rate of concentration increase in response to heat shock treatment. It's basically it. You just answer the question in one sentence. And for part B, I said when krill are placed in 4 degrees Celsius water, there's no change in concentration. But when they're placed in 10 degrees Celsius tanks, the concentration of mRNA-1 increases from 0 to 5 ml. When the krill are placed back into 4 degrees in the recovery period, the mRNA-1 concentration increases for the first 4 hours and then decreases for the last 2 hours. So basically for this question, you primarily refer to the graph and see how the graph trends for mRNA-1. And on the next slide, we'll be answering part C.
And just to add to what Samantha was saying, on the AP exam, they are going to try to give you these super long passages, and I kind of referenced it earlier, but you're able to answer these 2 questions really without having to read a whole bunch of stuff. So I really want you to focus on the models and graphs that you are given, okay? Take deep breaths. I know the reading is intense, but you can answer most of the questions from the graph. So like if you guys look, for what she did for letter B, she basically just described the graph. She just did it in words. And that's exactly what you guys should be doing, okay? So use the actual data, just like she did from 0 to 5, and she described it. It was absolutely perfect.
And yes, please do mention the specific concentrations. You do not want to lose points on the units. That's the easiest point to get. And here's part C. Yeah, for part B, you primarily describe what happens in the graph. In part C, the scientists hypothesized that the heat shock protein translated from mRNA-1 plays a greater role in refolding proteins than does the HSP translated from mRNA-2. Use the data to support the hypothesis. So for this question, let's spend 2 minutes on this question and begin. And that's time. Let's go look at the answer. So for part C, I answered it in this way. The graph shows that mRNA-1 concentration increases most drastically when the krill are moved back to 4 degrees Celsius water. This shows that after the krill proteins are denatured, mRNA-1 is transcribed at a greater frequency in order to refold the denatured proteins. Unlike mRNA-1, mRNA-2 has a steep decline in concentration when the krill are moved back to 4 degrees Celsius water, which shows that it's not involved in refolding denatured proteins. So basically for this question, they're asking which mRNA is mostly involved in refolding denatured proteins. And if you look at the graph, you can see that mRNA-1 denatures at much later time than mRNA-2. So it's most likely the mRNA that helps unfold or refold denatured proteins. And now on to our final part of the FRQ.
Really quickly, we have a question in the chat. So Samika, would you be able to go over what refolding is again?
Yeah. So basically for refolding proteins, they try to get back to the original shape. Yeah. So once an enzyme denatures, it won't fit into it won't allow the substrate to fit into the active site, right? So in order to by refolding denatured proteins, they're basically trying to fix their active site so that more substrates can be used in there. Yeah, and on to our final part. mRNA-1 and 2 are transcribed from the same gene. Explain how a cell can produce two different mRNAs from the same gene. And for this question, let's spend 2 minutes on it. And begin. And that's time. Let's go look at the answer. A cell can produce two different mRNAs from the same gene through alternative RNA splicing. Genes contain exons that alternate with non-coding regions called introns. When the transcription occurs, the pre-mRNA transcript with exons and introns and then has introns removed by a spliceosome, causing the exons to then be joined together. In alternative RNA splicing, different mRNAs can be produced depending on which regions are exons or introns. Yeah, I do want to reiterate for this part, there's many ways to answer it, but I believe the most Well, and personally, I believe the best way to answer is by talking about alternative RNA splicing. And that's all for the FRQ.
And then also to add to this, this is the genetics unit that we have. So in terms of the AP bio exam itself, each of the units are going to be weighted differently. So if I go back to the previous slides, sorry, bear with me as I get back to them. Each of, I guess I didn't have it on there, I apologize, but each of them are weighted differently. And so if you're sitting there trying to think about like what units should I focus on the most, the bigger units are going to be your genetics, evolution, and ecology. And so that's going to be the 3 main units on the test. So again, the bigger units are going to be ecology, evolution, and genetics. And so the FRQ that she just went over with you guys is in the genetics unit itself. It was kind of gene expression when you kind of went over operons, you went over transcription and translation. And so in terms of gene expression itself, if we're looking at a eukaryotic cell, remember that you're going to do transcription inside of the nucleus, and then you're going to translate it onto the ribosome itself. And so what we do is we edit the genes before they leave. And so what they're talking about right here is the alternative RNA splicing with the introns and the exons. And so those are kind of 2 keywords that you should know, introns versus exons. So in the chat, does anybody remember the difference between an intron and an exon? And I know my students better know it. Go ahead and chat. Tell me, define intron versus exon. See if you can type it in. Also, don't panic if not. There we go. Yash got it perfectly. Introns are going to be spliced out, okay? So intron versus exon. When you look at the DNA itself, so once we transcribe it, we are going to have parts of that we can kind of edit out and move around to make what we need. And so the parts that we don't need, we kind of refer to as like our junk DNA. Those are the introns. And if you guys look at the word intron, it has 2 N's in it. And so I tell my students to remember introns are not needed. Again, look at the word 2 N's, not needed. You guys are doing great in terms of your definition. Savannah, don't panic. We got this. Your exons are the part that you're going to want to keep. And I think about exons like I need exons for gas for my car, right? So I need the exons. And so you have these different enzymes called SNRPs. Hopefully you guys remember the name SNRPs. They're just these fancy enzymes. They're going to cut out the introns and then put together our mature mRNA strand. Okay, and so this question was really focusing on how do we get all the different genes from like a finite amount of DNA, which is kind of interesting. Okay, so transcription occurs in the nucleus, and then we're going to do a little editing, and then it's going to leave, and then it's going to go to a ribosome where we're going to translate it. So I guess my question for you guys is when thinking about translation on a ribosome, there are 2 types of ribosomes. You have ribosomes that are bound, and then you have some that are like free floating. Does anybody remember in the chat, what are my bound ribosomes bound to? What is that organelle that has all the ribosomes on it? Does anybody remember? It's a really long word. It's attached to the nucleus. What is the organelle? There it is, the rough ER. Great job, you guys. Look at that. Look, y'all are going to pass the exam. Easy peasy. Okay, so it is the rough ER. Very good. So the rough ER has the ribosomes on it. And so if our pre-mRNA, or really our mature RNA message now, is bound to a ribosome on the ER, then it's bound to leave. And that's how you remember that, okay? If it is a free-floating ribosome, then it's going to kind of stay inside of the actual cell itself. So bound ribosomes make proteins that are bound to leave, okay? So you have to kind of just remember that. If it is a prokaryotic cell, remember that we're going to kind of go through transcription and translation at the same time. They also have ribosomes, but they don't have those membrane-bound organelles like we do. So in terms of bacteria versus us, does anybody remember what are some organelles that we have that are also found in bacteria? Can anybody list any organelles that we have in common with bacteria? I know you can do it.
While they're typing in the chat, could we go over what exactly alternative splicing is and versus normal splicing? I've seen that question pop up a few times now.
It's the same. That is a very good question. So alternative splicing versus normal splicing. So when you are transcribing the message, so you have DNA, and then you go from DNA to your mRNA strand. Your DNA contains T's, so it's going to be like your ATCGs. And then when we're going to transcribe it, we're going to write the complement of the DNA strand, with the difference being in RNA, we use a special letter. Does anybody remember the letter you use in RNA? It's not T. What do we use in RNA? Do you guys remember from genetics? What does RNA use versus the letter T? There it is. Vivian got it. Rockstar, Hannah, Lexi, Rachel. All right, a U, very good. So in DNA, we're going to have the ATCGs, and then in RNA, we're going to have the U that gets paired with the A. And so what we'll do is before we leave the nucleus, we're going to have those SNRPs go through and cut out all those exons. And then we can rearrange the different parts of the RNA to make what we need. So when we say alternative splicing, it's the same thing as splicing. It's the same terms together. It just means how we can cut up the message and piece together what we actually need, okay? And so from there, I was looking at what you guys put in terms of the organelles that we had that were the same as bacteria, and you guys were right on. Okay, so let me go back to where you are. So we have ribosomes. We are going to have cytoplasm. We have cell membranes. We are going to have a nuclear region. Some of you guys put mitochondria, but mitochondria is a double membrane structure. So that's something that's only found in plants and animals. And so the other things that they have are sometimes they have a flagella, they have a cilia. They just don't have all the membrane-bound organelles that we do. And so I want you to kind of just remember that. In terms of genetic regulation, here I'm going to get a little deeper into genetics, okay? Does anybody remember the word that describes how bacteria organize all of their traits so that they can quickly respond to the environment? It starts with the O. Does anybody know that special word? It's like Jeopardy, essentially. How do bacteria organize their traits for gene regulation to quickly be able to respond to the environment when needed? There it is. There is Sarah, Rockstar, Adam, Ben. Great job, operons. Oh my goodness. Operons, great job. So operons are going to be how bacteria organize. Look at that. Y'all using those big science words. I'm very proud of you. That is exactly what they do. They're going to put all their genes that relate to one thing, so like metabolic, right? Whatever, metabolism, they put it all together. And so whenever they have all their genes together, upstream, they're going to have their operator, and then they're going to have their promoter, right? And so essentially, we're going to have a way of turning these operons on or off, okay? And that's how they're able to respond. And so the two that you needed to know were the lac operon and the trip operon, okay? So the lac operon is what bacteria are going to use when they want to break down the sugar in milk. Does anybody remember the name of the sugar in milk? I'll give you a hint. It's called the lac operon. What's the name of the sugar found in milk? It's a disaccharide. What's it going to break down? Lactose. Vivian, Arush, very good, you guys. Lactose, great job. Okay, it's a disaccharide. It's found in sugar. And our bacteria are going to use a lac operon to break down the lactose. Beautiful, okay? So right now, your bacteria is not going to have the lac operon on. So the lac operon starts off, okay? And so if I were to drink some milk, which I'm not, I have my water. If I were to drink milk, well, then we're going to go ahead and turn the operon on, okay? So the lac operon is going to start on off. And then when we're going to drink the milk, it's going to go ahead, turn on, and then break it down. Now, the other operon is called the tripopteron, and that operon makes tryptophan. So tryptophan is something that bacteria needs all the time. And so the tripopteron is always on, and then when we have too much, it's actually going to turn off the tripopteron, okay? And so that's called a feedback loop, essentially. Very good. And so that's kind of how our bacteria are going to control their genes, right? That's how they are very efficient in what they need to do to help to break down everything. And then we are going to go through transcription. We're going to edit our code before it leaves itself, and then we're going to translate it. Does anybody remember what proteins are made up of? There's like 20 of them. Starts with it, like 2 words. They both start with A. What are proteins made up of? What are proteins made up of? You guys, there it is, amino acids, and there's 20 of them, okay? My amino acids all have an amino group. They all have an acid group, okay? They all have a central carbon. What is the one part of amino acids that are different? Does anybody remember what that was called? What's the one thing that's different between all the amino acids? R groups, beautiful, very good. So the R group is going to be the one thing that's different between all of your amino acids, okay? And so that's kind of what gives those proteins those special properties. So the order of the amino acids is going to be very important in terms of the structure, and then the structure is going to be very important in terms of the function itself. So that's kind of a big theme that we have in AP Bio is structure determining function. Okay, so what is the term that we use to describe when an enzyme is broken down? Starts with a D. What happens when an enzyme is broken down? What's that scientific word? Starts with a D. It's what? Denatured. Chloe, first one. Great job, my love. Great job. Denatured. Okay, so when you have an FRQ that talks about enzymes, more than likely you have to talk about something being denatured. So don't say it's broken down. You're going to say it's denatured, okay? So enzymes are very specific in terms of the conditions that they prefer, okay? And so if you take an enzyme out of its optimal range, it's going to denature. So denature basically means you're going to break those bonds that keep that specific shape, because proteins have to be a specific shape. You change the shape, then you're going to go ahead and change the function itself, okay? So they have specific shapes. Denature means we're going to lose that shape, and now we can no longer do what we want to do, okay? So what are some environmental factors that can denature enzymes? What do you guys think? Can you put it in the chat? Again, what are some environmental factors that could denature an enzyme? Changes in what? Temperature. Beautiful. pH, salinity. Very good. Great job, you guys. Chemicals, yes. Very good. What are some non-environmental factors that could alter the shape of an enzyme? What are some non-environmental ones? What do you guys think? Besides temperature, pH, concentration. Very good, Becca. That's what I was looking for, okay? So concentration, right? Enzyme concentration versus substrate concentration, inhibitors. I view that was kind of that's right? So these are all inhibitors. Activators can also change those enzymes. Great job, you guys. So in terms of being able to describe what happens to enzymes, know that if you alter or change or have any of those factors, then it's going to break down those enzymes, okay? Enzymes can be reused, and if it's not too extreme, we can kind of refold and get those protein shapes back, and that's exactly what CER Method was talking about itself. Look at you guys using those keywords, allosteric inhibitors, beautiful, core pressors. You guys know a lot more than you think. You guys need to have confidence in yourselves. Beautiful. So the one thing that I want to mention that's kind of a misconception is that people will kind of say things to me like, the enzymes, if they get too hot, then they're going to break down. But I want you to kind of avoid saying vague things like that, because think about the enzymes that are actually in like hot springs. So enzymes have preferred conditions that they work best in, but like the enzymes in your stomach prefer the acidic environment versus like the enzymes at another place in your body, okay? So avoid saying vague things like if it's too hot, it's broke down. You would say that they have optimal conditions that they prefer, and then they are going to denature if not in those optimal conditions. All right, you guys have done great. You have like a little brief introduction to the science practices, okay? During our skills and drills night, for the first night, we're gonna do the first 3 practices, and then on the second night, we do the other 3. You know the format of your test. You know it's going to be 2 questions. You can do that, okay? And you know how to read error bars. So at this point, I'm going to kind of hand you over to Rashi.
So I've been seeing a lot in the chat about people being unsure. Oh, I'm going to get a 1 on this exam. I'm going to get a 2. I'm going to absolutely bomb it. I took AP Bio 2 years ago. And honestly, I can say that when I walked in for that exam, I felt like I knew nothing. I took it online. The class was at 8:30 a.m. So when I joined that Zoom, I was halfway falling asleep in the middle. And then the last 2 weeks before the actual exam, I was freaking out. I was like, oh my God, I knew nothing. And then I started cramming. And I'm sure many of you guys are in a similar spot today because your exam is like, what, in 1 week. But looking back, I can tell you that it was okay. I mean, I walked in there and I was nervous and my hands were shaking and they were sweating. And then I walked out of the exam and that was it. It was just an exam. At the end of the day, AP Bio is just a class and it is just an exam. And no matter what score you get on this, it does not define anything about yourself. It just defines, are you able to cram the AP Bio knowledge into your head, which isn't a measurement of how smart or talented you are. And the fact that you guys have shown up today for this formulas and graphing practice tells us all that you are willing to put in the effort to fully understand AP Bio to absolutely crush your exam. So you guys are going to be fine. No matter what score you get, we're all here at Fiveable. We're all super proud of you for making this effort and for joining this live stream. Okay, and we fully believe in you. You guys still have 1 more week, and that is plenty of time to study for this exam and absolutely crush it. So going over a few more last remarks. To recap this session, your exam is 2 parts. So there's 60 multiple choice and 6 FRQ questions. And it's tested on the application of knowledge and just not just the recall. So make sure that when you're studying, you're also quizzing yourself on how well you can apply the information. Because just if you haven't memorized, that won't be good enough, unfortunately. And it's assessed by these science practices. So we highly recommend you go back and check out the College Board list of science practices. For the math portion, double check that your calculator is approved and make sure you know what's on the formula sheet and what you have to memorize outside of that. TI-84s, by the way, are on the approved list. I saw a lot of questions on that. You guys can absolutely do this. You have all the resources you need, and we fully believe in you. And here at Fiveable, we have these cram sessions as well to help you out. So highly recommend signing up for these crams. I've used them multiple times, and every time I walk out of that cram session, I'm like, okay, I know what I'm doing now. I have a great starting point. I have a great cramming point. Fully recommend signing up for those. And the links have been dropped throughout the chats, so go check those out. Now, let's look at the actual exam statistics. If you look, you can see that 67.9% of students passed this exam. So about 70%. That's a lot of people. That's a very high passing average, which is why I know a lot of you guys are saying, I'm going to get a 1, I'm going to get a 2, it's going to be the end of the world. Odds are that you're going to pass. You have to keep that mentality, be confident in yourself, and believe in your efforts. And what? Oh, yes. And so I was also going to point out that look at the number of students who have taken this exam. Just last year, there were 240,000 students. That's a lot of people. You are being supported by a lot of people right now. There are a lot of other students taking this exam. So don't feel alone. I saw a lot of you guys talking about joining study sessions on Discord or reaching out to other people. And I highly recommend using those resources, using your fellow students, because they're in this boat with you. You are not alone for this exam. And so again, if you guys can see the link at the bottom, get your cram pass and study with us. Bubble has great resources. They have literally study guides for every single AP Bio individual unit. So if you were asking a lot of questions, like maybe you don't know the different types of inhibitors, then that's a great place to go and refresh up on that information. And AP Classroom is also incredibly helpful. A lot of you guys are also asking like, oh, is this answer good enough for the FRQ? Should I have included this information? AP Bio has practice FRQs, practice MCQs, and they have explanations for each question detailing why that answer is correct. But I also highly recommend you go check that out, because the more review you do, the easier it'll be to apply that information on the day of the actual exam. And so here is the rest of our cram schedule. So this is the second free preview. After this, you'll have to purchase an exam to watch the rest of the crams. So on the 5th, we'll go over units 1 to 4. On the 6th, 5 through 8. 7th and 8th will be skills and drills. So for those of you guys who are asking about MCQ practice and more FRQ practice, 7th and 8th is really like the best time, because we'll be doing all of that practice. We'll be going through the steps. And May 9th is our grand finale, one day before the exam. So please keep in mind your exam is on May 10th. It is at, I believe, noon, which means that there's over a week to study. And we highly recommend you guys come and study with us, because here we've all have experience taking AP Bio, and we all want to help make sure you guys do the best you can. And finally, make sure you follow at Think Fiveable on social media. We're on TikTok, Instagram, LinkedIn, YouTube. And our amazing social media team is phenomenal. I mean, I keep on seeing their TikToks and I laugh at them a lot, because I have been in that same boat about cramming for so many different AP exams at the last minute, so many different times. So go check it out. And please remember that we believe in you. You guys are going to kill this exam. And thank you so much for joining today.
Thank you so much, everybody, for joining us. If you could, in the chat, so we can end on a very good note, if you could leave us with a funny science joke, that would just allow us to breathe. Did anybody have any good science jokes that only us science people would understand? And if not, then I hope you guys have a fabulous night. If you want to rewatch this recording, once you go and click on the link that you clicked on to get here, the recording will show up like right away. All right, hope you guys have a great night. We look forward to seeing you. You can do this just like Rashi says. And if you could give us a good science joke so we can all end our night. Well, some of us are ending our night with a good science joke. We appreciate you guys.